great honor to present Bella CamachoHorvitz, who was working with me over those 8 weeks. So it went really fast because it was really a pleasure to have Bella in the lab. Like, she was always eager to learn new stuff and doing new techniques. She also came on a Saturday to do experiment, which is kind of amazing. So yeah, it was great. Thank you.
Okay, so the research that I did this summer was on mating behavior variabilities between Saccharomyces cerevisiae and Saccharomyces paradoxus. My mentor was Dr. and she was part of the Nathan Clark lab. So a little background on yeast. So yeast is one of the most commonly used model organisms for researching eukaryotic cells. And one of the reasons for why this is because they're able to reproduce either sexually as haploids or asexually as haploids or diploids. We really focus on the haploid cells because they can undergo sexual reproduction. And so in all yeast haploid cells, there's 2 mating types, and they're defined by alleles of their mat locus, either being mat A or mat alpha. So yeast that are mat A can only mate with yeast that are mat alpha, and the same thing goes with mat alpha and mat A and egg cells. So how they do this is that mat A, for example, in mat A cells, they produce this A factor pheromone, which is kind of like an invitation that they send to mat alpha cells, and it's they like to signal their presence, and they like to invite them and mate. So Saccharomyces cerevisiae, which is used to make bread and wine and beer, and Saccharomyces paradoxus, which is like a cousin of Cerevisiae. They're very similar, and they share the same genus and live in the same habitat, but are different species. So 2 organisms or species, if A, they're able to interbreed, and B, they're able to produce fertile offspring. And so Saccharomyces cerevisiae and Paradoxus, they are able to breed together, but all their offspring is infertile. So why do we care? So the big picture on this, which I didn't really get for a really long time, but Dr. Meslin gave me this great analogy of a horse and a donkey. So horses and donkeys, if they're crossed, they can produce an offspring of a mule, but the mule is sterile. So really, these 2, Paradoxus and Cerevisiae, they could be like a model organism for giving insights on bigger organisms like a horse and a donkey. So we focused a lot on the alpha factor pheromone, which is kind of the beginning of the mating pathway. And we realized that in Cerevisiae and Paradoxus, the sequence for the alpha factor pheromone is exactly the same. And that's odd because Cerevisiae has a very strong mating preference for mating with their own species, while Paradoxus doesn't really care whether it's a Cerevisiae or a Paradoxus cell that they mate with. So we kind of based our hypothesis on this fact, and we said that in Paradoxus, there's probably a mating deficiency that Cerevisiae can detect, and that's why they have a preference for their own species. So here's a visual of the STE2 cell receptor in mat A cells. And on the right here, there are cells of Cerevisiae and Paradoxus, but they're incapable of mating with other cells in this stage. But Cerevisiae and Paradoxus can both still undergo asexual reproduction. And here's a visual of the cell receptor when there's pheromone stimulation. And we focused a lot on the FUS3 protein because it really leads, when FUS3 is activated, it leads to phosphorylation of DIG1 and DIG2, which leads to the activation of STE12, which kind of gets the whole transcription of genes and is a really important factor in the mating pathway. And here are pictures of Cerevisiae and Paradoxus when there is stimulation. And you can see that they kind of look like pear shapes, and that means that they're schmooing, which means that the cells are getting ready for mating. So throughout my research, I really worked with 6 strains of Paradox, 3 of Paradoxus and 3 of Cerevisiae. And we got these from around the world, and they were engineered previously to be all MAT-A cell types. So I really wanted to answer the question, how do you detect the activation of mating pathways following pheromone exposure? So we did this in 3 methods. We took pictures of before and after pheromone stimulation. We did halo assay and a Western blot, which I'll explain later in the following slides. So to be able to detect any deficiency in the mating pathway, we performed this halo assay on all the strains. We did this by placing the alpha factor pheromone on these paper filters, and from there we placed them in a plate that contained a strain of yeast. And 24 hours later or so, if the yeast is able to detect the pheromone, they form this halo around the paper, and that means that their cell cycle was successfully stopped and there was no growth around the paper. So in our halo assay, we found that in both Paradoxus and Cerevisiae, there was pheromone detection. It's interesting to see that even in Paradoxus and even in their own species, for both species, there was a variability between how well they responded to the pheromone, which could have something to do with their environment that they were taken from, or other factors that we just don't know about. We did a Western blot to focus on the FUS3 protein, and if you remember, FUS3 is this part, which I talked about before. And so we found that we used 4 species in this one, and we saw that in both Paradoxus and Cerevisiae that there was FUS3 detection, which is shown by the bands in the FUS3 columns. And at 10 minutes, all were successfully phosphorylated. And you can see this on the right with this graph. And when there's a peak and when there's an increase, that means that there was detection of phosphorylation. Everything afterwards, there was a very small amount of time, and we didn't really have enough time to perform a lot of Western blots, so this was really the only one that I did. So we also, on the side, we had these wild yeast strains, and 2 samples were one from Pennsylvania and one from West Virginia. Those were the 2 that we did most of our experiments on. And so how we did that is we had these samples given to us by the Jen Gallagher lab, and from these solo samples, we added enrichment media that would take away all the bacteria from the tubes, and we started growing them, and you can see this white line is where the yeast is growing, and all the bubbles that are inside is where they're undergoing fermentation. And so we wanted to identify what these species of yeast were. So we ran a lot of PCR, and that was to amplify their DNA and be able to run a gel on them and be able to tell by their bands what type of species they were. So every 3 are a tree, and so the bottom bands are Cerevisiae, and we could, these are 2 of the same layout, and we both found them as Cerevisiae, and the top lane is Paradoxus. It was interesting to see, though, that when I wasn't so careful in getting a single cell, that we were able to find detections of both Cerevisiae and Paradoxus in a single tree, which means that even though but they weren't hybrids, so even though they were in a secluded environment and they were growing together and right next to each other, they still preferred to stay in their as their own Paradoxus or Cerevisiae separate species. In summary, Saccharomyces cerevisiae and Paradoxus strain, they exhibit different responses to the pheromone. And possible explanations for that is that there could be difference in the time that the 2 species sense the pheromone, or there could be lower activation of the mating pathway in one or the other. So from the lab strains that we had before, we want to be able to do the same thing that we did with from the wild yeast strains that we had before. We want to do the same thing that we did with the lab strains, and we want to be able to do halo assays and the Western blot, but we didn't have enough time to do that. So that would be our further direction, our future direction, to be able to investigate further on the main pathway of yeast. So I want to thank Dr. Camille Meslin and Nathan Clark, who is and all the members of the Clark Lab, because they really helped me a lot with everything that I did. Leah Russell, Sandy Yates, Dr. Yube, and the whole CSB department, Dr. Rose, Megan Houlihan, my DiSCoBio group members, UPCI, and my family.
So you have different levels of phosphorylation as a function of time in your Western blot.
Yes.
Do you have an explanation for that? Because that was very different between the different species. If you go back to your slide with the Western blot before there, so the phosphorylation, for instance, of fast 3 at 10 minutes is very strong, but at 30 minutes it disappears.
Well, yeah, like I said, after 10 minutes, things kind of got a little, swirly, and they started, something, probably my fault, happened, and the results didn't come out as they should have been, because the pheromone should have kept on going up, but we were really focused on the fact that at 10 minutes there was phosphorylation detection, which means that it was activated.
Do you expect to see a difference between the lab strains and the wild strains, or is there an advantage to using one over the other?
Well, the lab strains have been engineered a lot, and so I think that it would be really interesting to see how these yeast strains that we literally took out of the soil just under some trees, if there is a difference between after tons of engineering in the lab, if we could just straight off the bat just see if there's any difference, because we haven't really done anything to them other than performing a lot of PCR on them.
So are there, you go into the dirt, there's just two kinds of yeast, or there's lots and you just isolate those two types that were?
Well, yeah, so we started out using 6 primers of yeast, but we found that they weren't really showing up as any other type, and so we kind of focused on more Cerevisiae and Paradoxus, and we saw that there was a lot more Paradoxus than Cerevisiae, but there was actually one tree that we could just find nothing on, and so maybe that would be a different species. But yeah, I think that it depends where you're getting it from. I hear that in berries, they're mostly all Cerevisiae, but in trees, you can find a lot of Paradoxus Cerevisiae. Maybe in other environments, there are other Saccharomyces.